Hi there. In this video I'll be talking mainly about DNA replication, but I'll talk about some of the other molecules that are related to things like DNA polymerase, and then I'll discuss some details about DNA replication, including some of the different types of DNA replication. This diagram just shows you a bit about where we've been so far up here in general evolution, some of the pertinent molecules, especially nucleic acids, and then quite a bit about RNA, including ribosomal RNAs, tRNAs, mRNAs, and small RNAs, and then evolution of the genetic code and ribosomes. So now we're going to start talking about some specifics to DNA, starting with DNA replication, and then go to DNA segregation and mutation eventually. But this video is just on DNA replication. So DNA replication is performed by DNA polymerase, of course, but it still uses RNA primers, which tells you a bit about its history. So DNA polymerase is a DNA-dependent DNA polymerase, but there also are related molecules in the same family called DNA-dependent RNA polymerases, and those are the ones we talked about in the past few videos, including RNA polymerase, RNA pol1, pol2, pol3, and then I talked briefly about pol4 and pol5 as well. Also related to these are RNA-dependent DNA polymerases, and these are reverse transcriptases, telomerases, some transposons, etc. And then finally, these ones that are even further back in time as far as evolution are the RNA-dependent RNA polymerases, such as RNA replicase that's found in some viruses. And here's a general cladogram of the evolution of these different polymerases. So way back in the RNA world, you have RNA-dependent RNA polymerases only. And then coming out of the RNA world into a DNA world, then there's an RNA-dependent DNA polymerase, or reverse transcriptases, to make a DNA copy from an RNA template. From this also come both the DNA-dependent RNA polymerases and the DNA-dependent DNA polymerases, which are found in all cellular organisms, including all the way from bacteria to animals, plants, etc. Many of these also have telomerases, which are RNA-dependent DNA polymerases, so there are those also. But the central portions of those molecules, each one of those polymerases, has some similarities. So they're all evolved from a progenitor probably several billion years ago, at least 3.5 billion years ago. So here's a DNA molecule, and of course we talked about this earlier with regard to the structure of DNA, in that there's a 5' phosphate and the 5' pertaining to the 5' carbon on the deoxyribose sugar here. And then there's a 3' hydroxyl both on the end of this strand over here and this strand over here, and that's attached at the 3' carbon of the deoxyribose. And these are anti-parallel, but as you can see, if you know one strand, you can determine the complementary strand as well. Replicating DNA using DNA polymerase requires nucleoside triphosphates. That is, they have to have 3 phosphates on those nucleosides, otherwise they will not be added to the growing strand. And what happens is you have the 3' hydroxyl and phosphate groups coming together, forming an ester bond between them, joining up according to the complementarity of the bases. The other phosphates come off as a pyrophosphate, that is, two phosphates joined together without any carbon. If those build up, those inhibit the reaction. So there's actually a pyrophosphatase that recycles the phosphates. So there's always a template strand and a new strand being produced based on the complementarity of the bases to the nucleotides on the template strand. Polymerization usually begins in the middle somewhere of the molecule, not usually on the ends, and forms a replication bubble so that DNA polymerase and other proteins will sit in the middle of a replication origin of replications, and that will open up the two strands. DNA polymerase will begin synthesizing in the 5' to 3' direction from the template strand on both of the strands that are open. So on this blue one up here, you'll get polymerization, and on the red one down here, you have polymerization. To replicate the DNA or polymerize on that other part of the strand that's been opened up due to this leading strand, this continuous strand here, DNA polymerase again can only copy or polymerize from in a 5' to 3' direction. So essentially when this strand opens up, a DNA polymerase will sit on that and polymerize a small region until it joins up with the leading strand side. But then the end behind that is exposed, and so another DNA polymerase will pop back on that and synthesize another short fragment, and this continues on what's called the lagging strand. And this produces small fragments of DNA here called Okazaki fragments, named after the person who found them first and discovered that they were a natural part of DNA replication. Polymerization by DNA polymerase in a natural setting inside of a cell always begins from a short RNA primer, which is produced by a different enzyme. And then the DNA polymerase can pop onto that site and start copying from the template strand. When it meets up with the short fragment, it displaces those RNA fragments, and then ligase puts together the two pieces of DNA, the longer piece with one of the shorter pieces in the lagging strand. Several other proteins are involved here. So there is a topoisomerase, which will unwind the DNA ahead of that replication fork. There are single-stranded binding proteins that stabilize the single-stranded regions of the DNA, the single-strand DNA. And then there's helicase and other molecules that zip open the DNA so that the RNA polymerase can pop in there and move through to copy that strand of the DNA. You can also see the Okazaki fragments formed by the DNA polymerase, and then these green regions are the primers, the RNA primers that are needed so that the DNA polymerase can sit on there and start polymerization of the DNA strand. And building up a little more to show a few more details again, here's the topoisomerase, which is unwinding the DNA by cutting one or both strands and releasing some of the tension. There's helicase and single-stranded binding proteins, which are zipping open this DNA, the two strands, separating the two strands so that the DNA polymerase can pop in there. There's also a primosome that creates the RNAs that are the primers for the DNA polymerases, and then eventually there's a ligase that will put together seal the gap between those last two bases, between the longer piece of DNA in the lagging strand and the short Okazaki fragment. On the top here, this shows a few more details. Again, Here's the DNA polymerase on the leading strand. There's the one on the lagging strand. And note that they're in contact with one another. And so in vivo, this is also occurring. So the two DNA polymerase molecules and attendant proteins are actually attached to one another during replication. Here are the single-stranded binding proteins that stabilize the single-stranded DNA. There's the topoisomerase unwinding the DNA, the helicase, which is forcing those two strands apart. There's the primosome with the RNA primer that it's formed. And here is the DNA polymerase starting polymerization from one of those primers, and then eventually joining up with the longer piece on the lagging strand, and DNA ligase is sealing those gaps up again. So the leading strand and lagging strand. This shows a little more about how this is actually happening in a cell. So the DNA polymerases again are together, but while this DNA polymerase is polymerizing here, the DNA strands are being forced outward as the helicase splits those two pieces, the two strands. So this forms a little loop of DNA out here, single-stranded loop. Once the DNA polymerase then flips back onto the next primosome area with the primer, and this DNA is through, so it flips so that this little loop keeps getting longer, and then flips in and getting longer and flips in. So there's sort of a ratcheting effect as each one of the Okazaki fragments is formed. And the leading strand is pretty much smoothly going through, and there's no ratcheting. But these two molecules, the DNA polymerases, stay together during that time. So essentially you can look at this lagging strand as the polymerization is going forward, as the DNA is being separated here and moving out. The DNA polymerase then flips back onto the next primosome or primer, which releases this loop, and then it gets passed through, and then ligase seals those together, and then the DNA polymerase is back on the next section, starts copying that as the DNA is also, the single-stranded DNA is being looped out again. Once it finishes, it flips back and lets go of that part, and then the DNA ligase seals that, etc. So it's sort of, the loop comes out, flips in, and the loop comes out, flips in again. So usually people are talking about replication of a bacterial chromosome. So then you replicate it. There are two chromosomes, the cell divides, and then the daughter cells each have a copy of the chromosome. Or in a diploid cell, you have pairs of chromosomes. Those get replicated and then separated during mitosis or meiosis, depending on what cells they are, and each cell then gets an equal amount of the DNA. But I want to introduce some other things that happen too, and then some different types of replication. So I need to talk a little bit about C numbers, that is the haploid amounts of DNA or the genome, and N is the chromosome number, because those can be different, and you can have different amounts of DNA and different numbers of chromosomes moving around in those cells. So 1C equals the haploid amount of a DNA, usually referred to as the genome also. 2C is the diploid amount, 3C triploid, 4C tetraploid, and then multiples of C, like 90ploid is 90 copies of the genome, etc. Because chromosomes sometimes are held together for long periods of time, even after replication, then the chromosome numbers that you can count in the microscope can differ from the amount of DNA in a cell. So in humans, the 1N number, the number of chromosomes, is 23, so 2N would be 46 chromosomes per diploid nucleus. In mice, 1N equals 20, in broadbean, 1N equals 6 per haploid nucleus in each case. So in a typical bacterial cell cycle, starting here with a 1C haploid, essentially haploid amount of DNA in G1, some bacteria can differentiate, some can essentially be resting in a spore or something like that. And then you go into the S phase where the amount of DNA increases from a 1C level to a 2C level. And then there's a stage here called G2 where the cells have doubled their chromosomes, so they have or doubled their genome essentially, and so they have twice the amount of DNA that cells over here do. And sometimes you can have a resting stage here and then go through binary fission, separating the two chromosomes equally among the two daughter cells. And if you look at the amount of DNA in an E. coli cell, it looks actually impossible that amount of DNA could fit into that little cell. This is a prep where they've sort of opened up pores in the E. coli cell and allowed it to spill its DNA out. This diagram's kind of misleading because what they've done also is they have added certain proteins to the DNA and then shadowed the DNA or stained it with a combination of heavy metals, usually uranium, platinum, palladium, things like that, to make them visible. So you need something that's electron dense in order to see it in the microscope, and DNA isn't very electron dense. So you add things onto the DNA that make it electron dense, and so that makes the DNA much thicker. So the DNA that's actually shown here has a coating of those heavy metals around it, or heavy atoms. So it looks much thicker. It's actually much thinner than that, and all of that DNA does actually fit into that bacterial cell. Nonetheless, that is a lot of DNA to replicate, and to replicate accurately, and to divide equally between two daughter cells. So it's quite a feat to replicate all that DNA, separate it when the cells are dividing, and so that there aren't broken pieces of DNA or pieces of DNA that are linked together in loops and you can't get them apart. I always tell students to take 2 balls of string of different colors and take that yarn or string out and mix it up in a bag, pull it back out, and then try and separate it. It really gets tangled in a hurry. So bacteria and archaea normally have circular chromosomes, although there's some discrepancy about that, some question about that. But anyway, so you could take all that jumble of DNA that I showed in the previous slide, and if you spread it out on a table somewhere, it would be a circle. But these, as I said, start from a replication bubble at an origin of replication. You have a leading strand going around the DNA, copying one strand, and then a lagging strand going and copying the other strand of the DNA, eventually coming out with 2 new circles, one containing the new strand on the inside here and the old strand on the outside, and the other one having the new strand on the outside here and the old strand on the inside here. So they can be slightly different molecules or slightly different versions of the genome. So they replicate in a form called theta, because the molecules, while they're replicating, look like the Greek symbol theta. And this is one of the famous figures from John Cairns, who discovered how DNA replicates in a circular molecule in bacteria from E. coli, actually. And he used radioactive nucleotides and then isolated replicating molecules, put them on some photographic film, and looked at where the radiation was exposing the film, and could follow the replication of these molecules. So there's a replication fork here, a replication fork here. So this is the original molecule here, and then these two little ears coming out here are the newly synthesized DNA copies. But there are other versions of DNA replication. One is called rolling circle replication, used by some viruses, including lambda, viroids, and some other systems in other organisms. So replication starts from a certain point where there is a specific sequence in the DNA. This is nicked by an enzyme that cuts only one strand. So now you have a nicked circular piece of DNA. Next, DNA polymerase sits on that site and starts replicating the DNA in one direction. And in doing so, it displaces the other strand, which will become the lagging strand. But the one going around the circle will be the leading strand of the replication. Once the displaced strand is long enough for DNA polymerase to pop onto that site, it does, starts polymerizing the part that's exposed, and then pops over to polymerize the next section and the next section, just as in any DNA replication process. And so this produces one continuous strand here on the leading, which is the leading strand, and then pieces joined together on the lagging strand, eventually joining everything up. And if you look at the point where the nick occurred, then you have multiple end-to-end concatenamers, that is, genomes worth of DNA joined end to end to end. So it forms long, continuous pieces of DNA containing multiple versions of the genome of that organism, such as lambda. Another form of replication is called recombination-dependent replication, or RDR. This occurs in some viruses, some organelles, and it may occur with bacterial chromosomes as well. There are some bacteria also that use this system, especially things where their DNAs are often broken up, such as Deinococcus, which can withstand high doses of radiation. So anyway, you have sections of chromosome separated from one another to start out with. And this may seem somewhat odd because you think it should be a circle or a chromosome, but this is actually quite common in some viruses, in organelles, and in some bacteria. Because these pieces of chromosome are homologous along their lengths, then one strand can actually invade the other strand and hybridize with it because it has the same sequence, so they are hybridizing just the way the other strand would. But you get strand invasion and then a loop out called a D-loop on this other strand up down here. Next, DNA polymerase can pop onto that site and begin replicating that strand from the template on the bottom. And then it leaves this single-stranded part of this DNA looped out in, again, what's called a D-loop, because in the microscope it looks like the letter D. And lagging strand replication can then begin on that displaced loop. So now you're getting replication on both of those strands to make new portions of those chromosomes. And eventually, after everything is copied, you still have a crossover in the middle, which was initiated by this strand invasion up on the top here. The crossover can be resolved in two different ways. One is that the crossover there can be clipped. DNA ligase can then join those pieces together, those nicks together, into two long chromosome pieces. or you can cut across just one of the strands or the two strands this way and not ligate them together, and you get separated chromosomes similar to what you started with up here. But if you'll notice on this one that's been joined together, there's a heteroduplexed region. Now when I start talking about genetic recombination and crossovers, I'll be talking in more detail about these heteroduplexed regions. So it really is a consequence of both recombination and replication. Now, as I said, this type of replication, RDR, has been found in some organelles, and it was found during some mapping projects trying to map out plant mitochondrial DNA, what the chromosome looked like. And actually I was involved in early parts of this. We were trying to find in the electron microscope large circles, the genomic circle of a mitochondrial DNA in a plant. And every time we'd look in the microscope, the electron microscope, we never got large circles. Sometimes we got small circles that weren't large enough to be the genome, but most of the time we just got a set of linears. And we thought we were doing something wrong, but actually when we look back on it, that's what you find in mitochondria, a bunch of linear, seemingly random sized pieces of linear DNA. And this was an early indication that these mitochondrial genomes were replicating by RDR. And so here, if you have these genomes of plant mitochondrial DNA, just various pieces, and you get a crossover here to initiate replication. And then you go through the replication process, and now you have one longer molecule that has double the number of genes, essentially, and then a small piece that is similar to the starting fragments. But RDR doesn't stop there. You have additional rounds of recombination-dependent replication, and you get these long branched molecules as well as some small molecules. This kind of thing is also found in certain viruses, so it's not uncommon in different types of organisms. It may occur in also chloroplast DNA and bacteria, some bacteria. So it seems that this type of replication has been successful, although it looks a bit confusing when you first view it. So eventually, when this replication is all completed, you get resolution of these different crossovers. What happens is you find the DNA is fragmented. So all of the genes are there, and sometimes multiple copies of the genes are there. But all of the genes are within that organelle, or within that bacterium, or within that virus, but they're in essentially random-sized pieces. So when we did our preps of mitochondrial DNA, looked in the microscope, and we were trying to find circular molecules, and we looked for days and days and weeks for these large genome size molecules. Never found them, just never found them. We found only pieces, randomly sized pieces of DNA, and lots of them. So we had the DNA. We thought we were doing something wrong. For some reason, we thought the DNA was being digested, had no idea. In reality, we were seeing reality. We were seeing what actually was in the mitochondria, and other subsequent experiments have shown that there are no large genome size chromosomes or genomes in these plant mitochondria. But the genome is fragmented like this, which indicates that it's being replicated by RDR. The curious thing, though, is that if you do sequencing and mapping and have the computer or your friend join these pieces together, because there are overlaps in these, right? So you can find the overlaps And when you or the computer lines them all up, a circular type map of the genome can be constructed. So out there in the literature, you find mitochondrial maps that have been published that are circular. And they may or may not be, although the plant mitochondrial DNAs and the plant mitochondria that we looked at, yeah, you could form a circular map based on the sequences and the restriction digests and things like that. But when that was studied even more closely in the organism, there were no circular molecules. The genome was fragmented, as you can see over here on the left in this model. So some of these maps that have been published that look like the mitochondrial genomes and the bacterial genomes are circles. Maybe these only exist on paper, and actually when you go into the organism, those genomes are not circular, but they're fragmented, partly because of the way that these organisms replicate their DNA, and possibly for other reasons as well. Okay, so that's just the beginning of our journey through DNA, how it replicates, and we'll talk about how the DNA is segregated, that is by fission, by mitosis, by meiosis, and other means in the upcoming videos. So today I just talked about standard ways of replicating DNA, rolling circle, as well as this RDR method of replicating DNA. So that's it for now. Thanks for watching and listening, and I will talk to you later.